Assalamu alaikum, dear students. My name is Dr. Mohammad Latif, and I'm your instructor of the subject Principle of Animal Life 2. Today is the lecture number 11, in which we will talk about linkage. This lecture and the coming lecture, which is lecture 12, is basically both cover this topic, which is linkage. So after these two lectures, you will be able to understand what is linkage is. So you must familiar with the genetic map. What is genetic map? Genetic map, basically it is a diagram that describes the order of genes along a chromosome. You will see, you have all familiar about chromosome, that every species there is a specific number of chromosomes, and chromosomes basically are made from DNA and histone proteins, and in DNA, which has DNA is the genetic material which has all the information of the proteins or all the characters. So DNA has basic functional unit which is called genes, and basically genes influence an organism traits because genes are involved in controlling different traits in an organism. And you must also knows about that there is a fixed number of chromosome in every individual or in every species. I will talk about humans. Humans have 46 chromosomes, but the number of genes present in humans are more than 30,000. So a typical chromosome is expected to contain many hundreds or perhaps a few thousand different genes, because there is only 46 chromosomes, but there are thousands of genes. So a single chromosome can contain many hundreds or perhaps a few thousand different genes. And what is genetic linkage or simply linkage? Basically, it is the phenomena in which genes that are close together on the same chromosome tend to be transmitted as a unit, influence on inheritance pattern. इसका मतलब क्या है कि लिंकेज वो फिनोमिना है कि वो वाले जीन्स जो एक दूसरे के बहुत ही करीब होते हैं क्रोमोसोम्स के ऊपर या एक दूसरे के बिल्कुल साथ मौजूद होते हैं, वो एज ए यूनिट अगले जो ऑफस्प्रिंग हैं, जो उसके ऑर्गेनिज्म की जो आने वाली स्पीशीज हैं उसके अंदर वो इकट्ठे यूनिट के तौर पे ही ट्रांसमिट होते हैं. Or chromosomes are sometimes called linkage groups because a chromosome contains a group of genes that are physically linked together. And in each species, the number of linkage groups equals the number of chromosome types. For example, human somatic cells have 46 chromosomes, in which 22 are autosomal linkage groups and X chromosome linkage group, and in males have a Y chromosome linkage group. And in addition, I'm talking about a gamete, which when we call gamete have 22 autosomal linkage and one X chromosome in case of female, and in case of male it is Y chromosome. And in addition, there is a human mitochondrial genome is another linkage group. And the outcome of a dihybrid or a trihybrid cross depends on whether or not the genes are linked to each other along the same chromosome. जो हमने डाईहाइब्रिड और ट्राईहाइब्रिड क्रॉस हमने पढ़ी थी, जिसके अंदर 2 और 3 कैरेक्टर्स एट ए टाइम हम किसी भी इंडिविजुअल है उसके अंदर देखते हैं कि 2 क्रॉस यानी 2 कैरेक्टर एक साथ जब किस तरीके से F1 यानी फर्स्ट फीलियल जनरेशन के अंदर और सेकंड फीलियल जनरेशन के अंदर वो ट्रांसमिट होते हैं। तो जब लिंकेज फिनोमेना आएगा इसके अंदर इनका बहुत ही ज़्यादा इम्पोर्टेंट फिनोमेना मतलब बनेगा। क्यों? क्योंकि जो नॉर्मल मेंडेलियन रेशियो होती है जो 9331 है, जिन जीन्स के अंदर लिंकेजेस मौजूद होता है तो वो वाले ट्रेड्स जो होते हैं उनके अंदर जो हमारे रिजल्ट्स आते हैं वो डिफरेंट होते हैं फ्रॉम 9331, जो एक नॉर्मल मेंडेलियन रेशियो है जो आपने लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट में आपने पढ़ी थी कि जो 9331 रेशियो है ये उस वक्त आएगी जब लिंकेज अवेलेबल नहीं होगा। अगर लिंकेज होगा तो ये जो 9331 रेशियो है ये आप अचीव नहीं कर सकेंगे। देयर इज अ डिफरेंस ऑफ रेशियो ऑफ मेंडेलियन जो हमारे लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट में आएगी। अच्छा, Bateson and Punnet discovered 2 traits that did not assort independently. जो some of the traits may not assort independently was carried out by William Bateson and Reginald Punnet in 1905. And Mendel's law dihybrid cross, the ratio was 9331 as I already mentioned in my previous slides. And Bateson and Punnet cross a true breeding strain with purple flowers and long pollen to a strain with red flowers and round pollen. And in F1 generation, the plants that had purple flowers and long pollen are achieved. Means the trait of purple flowers and long pollen are dominant over a trait of red flowers and round pollen. But in F2 generation had a much greater proportion of the 2 phenotypes found in the parental generation. What is parental generation? Phenotypes are: one parental generation is called purple flowers with long pollen and the other parental strain are red flower with round pollen. Actually, the ratio must be 9331, which is the normal Mendelian ratio. But these two scientists found a different ratio, and in which the different ratio is this, that the parental generation have much greater proportion of the 2 phenotypes, and therefore they suggested that the transmission of these 2 traits from the parental generation to the F2 generation was somehow coupled and not easily assorted in an independent manner, because they do not have any knowledge about the linkage phenomena, because Bateson and Punnet did not realize that this coupling was due to the linkage of the flower color gene and the pollen shaped gene on the same chromosome, because the flower color and the pollen shape They are basically present on the same chromosome and they are closely linked to each other, and at that time the linkage phenomena is not fully studied. This is the cross between two parental generations. One is purple flowers with long pollen and red flowers with round pollen. And with F1 generation, all are purple flowers with long pollen. And when it is self-fertilized, the F2 generation, different results are obtained. Purple flowers, long pollen, these are the parental generation. 296 and the ratio is 15.6. The normal expected number is 240, which is a ratio 9. Purple flowers, round pollen. This is a new combination and this is much less. The normal expected ratio was 3, but in actual ratio is only 1. The red flowers, long pollen, this is again a new combination, or again the ratio is much less than the normal, which is 3, 1.4. And other is the red flowers and round pollen, which is a second parental generation, which is recessive to the purple and long pollen. Its normal ratio is 1, but the actual ratio was 4.5. So what is the relationship between linkage and crossing over? Even though the alleles for different genes may be linked along the same chromosome, and the linkage can be altered during meiosis. In diploid eukaryotic species, homologous chromosomes can exchange pieces with each other, a phenomena called crossing over. हमने ये completely आपने को क्रॉसिंग ओवर का फिनोमेना मैंने समझाया जब मैंने आपको मियोसिस बताई थी, और मियोसिस के अंदर हमने देखा था कि जो मियोटिक 1 है उसकी प्रोफेज 1 के अंदर there is a exchange of segments, और इन exchange of segments को हम क्या कहते हैं, crossing over. And this event occurs during prophase 1 of meiosis 1. And the replicated chromosome known as sister chromatids associate with the homologous sister chromatids to form a structure known as bivalent. Bivalent means दो homologous chromosome क्या है, ये भी मैंने आपको बताया था कि वो क्रोमोसोम जो साइज और शेप और नंबर ऑफ जीन्स के हवाले से एक जैसे हों, यानी जिनके अंदर नंबर ऑफ जीन्स, साइज और उनकी जो शेप है वो सेम होगी, ऐसे क्रोमोसोम को हम होमोलोगस क्रोमोसोम कहते हैं. जैसे हम कहते हैं हमारे यहाँ 46 क्रोमोसोम है, तो देयर आर 23 पेयर ऑफ होमोलोगस क्रोमोसोम, यानी उसमें 23 अगर हम बनाएंगे तो पेयर बनते हैं, यानी उसके अंदर हर क्रोमोसोम अपने जैसा एक क्रोमोसोम है जिसके अंदर उसके साइज भी सेम है, शेप भी सेम है, और तो जीन्स जो दोनों जो मौजूद हैं वो भी दोनों के ऊपर सेम है. और दैट्स वाई जब वो अलहदा होते हैं 23 यानी एक दूसरे से अलहदा हुए, और अपोजिट पार्टनर जब यानी दूसरा गैमेट इनके साथ मिलता है उनके पास भी वो सेम होते हैं, तो इस तरीके से वो आगे जनरेशन टू जनरेशन ट्रांसफ़र होते हैं. और जब दो होमोलोगस क्रोमोसोम एक दूसरे के करीब आएंगे ड्यूरिंग क्रॉसिंग ओवर, तो क्योंकि दोनों क्रोमोसोम डुप्लीकेटेड हैं, इसके ऊपर भी 2 क्रोमेटिड्स होंगे, उसके ऊपर भी 2 क्रोमेटिड्स होते, तो इसको हम टेट्राड कहते हैं. ये भी मैंने प्रोसेस आपको समझाया था, यू मस्ट फेमिलियर विद टेट्राड. और टेट्राड क्या होते हैं कि जो 2 पेयर्स ऑफ सिस्टर क्रोमेटिड्स एक दूसरे के साथ आ जाते हैं, और फिर ये दोनों सिस्टर क्रोमेटिड्स जो होते हैं वो अपने एक्सचेंज ऑफ सेगमेंट्स करते हैं। क्रॉसिंग ओवर may produce recombinant genotypes। जो हम वी आर मस्ट नो अबाउट क्रॉसिंग ओवर के क्रॉसिंग ओवर से होता क्या है, इससे न्यू कॉम्बिनेशन बनते हैं, और अगर क्रॉसिंग ओवर नहीं होती तो कोई नया कॉम्बिनेशन नहीं बनेगा। और आगे फिगर आपको बताएगी कि मियोसिस के अंदर जब टू जीन्स आर लिंक्ड ऑन द सेम क्रोमोसोम, नो क्रॉसिंग ओवर हैज अकर्ड। When genes are linked, the phenomena of crossing over cannot occur. Therefore, the resulting haploid cells contain the same combination of alleles as the original chromosome. यानी जो subsequent जो इसका बनेगा गैमेट, उसके अंदर अगर क्रॉसिंग ओवर नहीं हुई तो जो अलील पेरेंट के अंदर मौजूद था वही आगे ऑफस्प्रिंग के अंदर आ गया। इन दिस केस टू हप्लॉइड सेल्स कैरी द डोमिनेंट। जैसे फॉर एग्जांपल अगर हम कहते हैं कि एक अलील है जिसके अंदर दोनों डोमिनेंट अलील मौजूद हैं, जिसको हमने डिजिग्नेट किया है विद ए एंड बी अलील्स, और जो इसका रिसेसिव जो दूसरा क्रोमोसोम है उसके बाद दोनों रिसेसिव अलील हैं जिसको हमने स्मॉल ए और स्मॉल बी, ये देखें हमने डोमिनेंट को कैपिटल ए कैपिटल बी और इसको स्मॉल को हमने, ये इसके जो अदर पार्टनर है उसको हमने स्मॉल ए और स्मॉल बी के साथ डिजिग्नेट किया। दीज आर कॉल्ड नॉन रिकम्बिनेंट सेल्स। नॉन रिकम्बिनेंट इसलिए कहा क्योंकि आपस में इन्होंने कोई एक्सचेंज ऑफ एक्सचेंज ऑफ सेगमेंट नहीं किया। अगर एक्सचेंज ऑफ सेगमेंट किया होता तो ये आपस में एक दूसरे के साथ क्रॉस होते, तो मे बी जो अदर जितने भी आगे गैमेट्स बनते उनमें एक डोमिनेंट वाला होता और एक रिसेसिव वाला होता। ठीक है, ये वाला कॉम्बिनेशन बन सकता था, ये वाला कॉम्बिनेशन बन सकता था, यानी बी ए के साथ आ सकता था और ए बी के साथ आ सकता था, क्योंकि अगर वो क्रॉसिंग ओवर हुई होती। लेकिन अगर क्रॉसिंग ओवर नहीं है तो ये सेम ही अलील एक जगह पे मौजूद होगा और सेम अलील दूसरी जगह पे मौजूद होगा। These are called non-recombinant cells or parental cells. The arrangement of linked alleles has not been altered from those found in the original cell. लेकिन नेक्स्ट फिगर में मैं आपको बताऊंगा what can happen when crossing over occurs। यानी एक केस ये है कि जिसके अंदर क्रॉसिंग ओवर नहीं हुई, and the other case in which crossing over occurs. So the resultant जो haploid gametes they are altogether different from the parents because it has both the alleles, one with dominant and other with recessive, and other one is recessive and dominant. So these are called a new combination of alleles, और इसको हम genetic recombination कहते हैं। The haploid cells carry the capital A and small b, or the small b and capital B alleles are called recombinant cells or non-parental cells. So when these gametes जब नए ऑफस्प्रिंग्स के अंदर जाएंगे तो इनके जो कैरेक्टर्स हैं दे आर डिफरेंट फ्रॉम देयर पेरेंट्स। और likewise if such haploid cells were gametes they participate in fertilization, the resulting offsprings are called recombinant offsprings और नॉन पेरेंटल ऑफस्प्रिंग। जैसे कि नॉर्मली हम देखते हैं जब we see the dihybrid class in law of independent assortment, we have seen that there are different combination of offspring which are appear in F2 generation, and the formation of these new combination is due to the crossing over phenomena. So in both we have in this picture the both scenario. The one scenario is without crossing over and the other scenario is with crossing over. And when a diploid cell after chromosome replication, and there is no crossing over because the both alleles are linked to each other, you will see that they have the same allele which are present on the parent, and it is transmitted in their gametes. The possible haploid cells have the same combination which the parent have. You must know that these are duplicated chromosome, but in gametes they contain a single copy, and so the single copy have the same alleles which are present in parent. But what happened when crossing over took place? And when crossing over took place, there is a different combination in possible haploid cells. So we have variety B with A or B with small a means recessive of this, and small b with small a or small b with capital A. So all possible combinations. And capital B can be with capital A, capital B can be with small a, or small b can be with small a, or small b can be with capital A. So these are the all four combination if there is a crossing over take place.
Linked genes are not independently inherited. For an example, let's imagine a Mendelian dihybrid cross using fruit flies. The male on the left has a recessive black body trait and recessive purple eye trait. The female on the right has the dominant caramel body color trait and dominant red eye color trait. Since this is a Mendelian cross, we can assume they are homozygous for both traits. In this instance, the letter E represents eye color and the letter B represents body color. Linked genes are by definition on the same chromosome, so it's helpful to imagine the genes as they occur in nature, that is, on a chromosome. A fruit fly karyotype would have 4 pairs of chromosomes. The black body and purple eye genes are on chromosome 2, so we will only consider that pair. Notice that the eye color and body color genes are represented as an integral part of our diagrammatic chromosomes. The original mating pair, or P1 generation, are homozygous for both traits. So after meiosis, each will donate 1 copy of the chromosome pair to any progeny. The progeny, or F1 generation, will be heterozygous for both traits and will all have the dominant set of phenotypes. Now let's set up the F1 cross. If linkage is being tested, then the F1 cross will not follow the standard Mendelian scheme. Instead, the F1 progeny will be mated to a homozygous recessive, similar to a test cross. In this scenario, the homozygous F1 parent can only donate recessive traits to its offspring. That way, any differences we see in the F2 generation will be the result of meiotic events from the heterozygous parent. If that parent passes along the chromosome with both recessive traits, then the F2 individual produced from that fusion will be genotypically homozygous recessive, phenotypically recessive for both traits, and will resemble one of the original parents. We would call that phenotype parental recessive. If that parent passes along the chromosome with both dominant traits, then the F2 individual produced from that fusion will be genotypically heterozygous, phenotypically dominant for both traits, and will resemble one of the original parents. We would call that phenotype parental dominant. Recall that during meiosis, crossing over can occur between homologous pairs of chromosomes. If a crossover event occurs between the 2 genes responsible for the traits we are following, then the combination of traits will change. Now it is possible that an F2 progeny could be dominant for eye color, yet recessive for body color. This combination is called non-parental because it did not occur in either of the original parents in the P1 generation. Conversely, the F2 progeny could be recessive for eye color and dominant for body color. This combination is also non-parental. When genes are linked, the proportion of each phenotype is determined by the distance between the two genes. For example, if the black body and purple eye traits are very close to one another, we would expect very few non-parentals compared to parentals. That's because the likelihood of a crossover occurring within that small area on the chromosome is also small. The likelihood of a crossover between the genes increases with distance. So the farther any two linked genes are from one another, the greater the number of non-parentals compared to parentals. Since there is a direct correlation between gene proximity and non-parentals, we can use the proportion of non-parentals to parentals to calculate an approximate distance between two linked genes. Let's say we count the number of parentals versus non-parentals in our example cross. We might count 94 parentals and 6 non-parentals. Therefore, 6% of the total number of F2 flies are non-parentals. Geneticists call this percentage a genetic map distance and would describe the two genes as being 6 map units or centimorgans apart.